Thank you. Well, full story report of andexanet alpha for bleeding associated with factor Xa inhibitors. First, we have to talk about the methods. The methods. We evaluated 352 patients who had an acute major bleeding within 18 hours after administration of a factor Xa inhibitor. The patient received a bolus of andexanet followed by 2 hours infusion. The co-primary outcomes were the percent change in anti-factor Xa activity after andexanet treatment and the percentage of patients were excellent or good hemostatic efficacy of 12 hours after the end of the infusion. We divided the methods in 4 steps. The first steps were the study design and oversight. Study design and oversight. This was a multi-center perspective open-label single-group study. The Population Health Research Institute (PHRI) at McMaster University and the industry sponsor Portola Pharmaceuticals jointly designing the study, and both selected sites and supervised monitoring. The protocol, consent forms, and ancillary materials were approved by institutional review boards at each center. The next step was study population. The study population patients were enrolled at 63 centers in North America and Europe. Patients were eligible if they were at least 18 years of age, presented with an acute major bleeding, and had received within 18 hours on the following apixaban, rivaroxaban, and edoxaban at any dose, or enoxaparin at a dose of at least 1 mg per kilogram of body weight per day. The next step of the methods were study procedures and data collection. Eligible consenting patients receive an andexanet bolus over a period of 15 to 30 minutes followed by 2 hours of infusion of the drug. The following doses were used in the initial protocol for all patients who had received apixaban and those who had received rivaroxaban more than 7 hours before bolus administration. The bolus dose were 400 mg over a period of 15 minutes, and the infusion dose was already 480 mg. The next step was the study outcomes. The study outcome has 2 primary efficacy outcomes: the percent change from baseline in anti-factor Xa activity after andexanet treatment, and the percentage of patients with excellent or good hemostatic efficacy 12 hours after the andexanet infusion, with hemostatic efficacy assessed by an independent education committee on the basis of perspective criteria. Information regarding change of the primary outcome during the trial and details concerning the adjudication of hemostatic efficacy are provided in the supplementary. The next step was the results. Patients has a new age of 77 years and must have substantial cardiovascular diseases. Bleeding was predominantly intracranial in 227 patients, or 64%, or gastrointestinal in 20 patients, 26%. In patients who have received apixaban, the median anti-factor Xa activity decreased from 149.7 mg per millimeter at baseline to 11.1 mg per millimeter after the andexanet bolus. 92% reduction to 95%. Confidence interval 91 to 93 in patients who have received rivaroxaban. We divided the results in 3 steps. The patients we enrolled 352 patients from April 2005 to May 2018, including the 67 patients reported previously. All the patients received andexanet and were followed for at least 30 days or until death. Patients had a mean age of 77 years. Baffling medical history included myocardial infarction in 48 patients, 14%, strike in 79, 20%, and deep vein thrombosis in 77 patients, 19%. Table of atrial fibrillation was the primary indication for anticoagulation in 280 patients. The next step was the antifactory activity. Antifactory activity. The antifactor activity, antifactor 10A activity in the efficacy population among the 132 patients who were receiving a Pixelban, the median value for antifactor 10A activity was reduced from 149.7 milligrams per milliliter at baseline to 11.1 milligrams per milliliter at the end of the bolus administration at 92% reduction, 95 confidence interval among the 100 patients who were receiving rivaroxaban. The median value for antifactor 10A activity fell from 211.8 milligrams per milliliter at baseline to 14.2 milligrams per milliliter at the end of the bolus administration of 92 reduction percent. The next step was hemostatic efficacy. Of the 254 patients in the efficacy analysis, 249 could be evaluated for hemostatic efficacy, and 204 were adjudicated as having excellent or good hemostatic efficacy at 12 hours. Of these, 171 were adjudicated as having excellent hemostatic efficacy and 33 as having good hemostatic efficacy. The percentage of patients with excellent or good efficacy were 85% for gastrointestinal bleeding and 80% for intracranial bleeding. And finally, we have the conclusions. The conclusion that we get from this study was that in patients with an acute measure of bleeding associated with the use of a factor 10A inhibitor, treatment with andexanet markedly reduced antifactor 10A activity, and 82% of the patients had an excellent and good hemostatic efficacy at 12 hours, as adjudicated according to the specified criteria. And finally, we have the videos for more